Hello again, everybody. Welcome to the webinar on Steps for Success with the RNAscope Multiplex Fluorescent Assay. My name is Sudha Krishnan, Technical Support Specialist, and I'll be discussing some key pointers and suggestions for a successful RNAscope Multiplex Fluorescent Assay. A few pointers about the webinar. The audience will automatically be muted. If you would like to ask questions, please select the chat button to type in your questions. If we are unable to answer your questions, we will follow up with you offline. For today's webinar, we'll start off with a brief introduction to RNAscope in general. Within this segment, I will be going over the RNAscope workflow and some tips and suggestions for the RNAscope Multiplex Fluorescent Assay, and we'll finally be touching up on specific troubleshooting tips for the assay. We will then wrap up with a question and answer session. I would like to mention here that this webinar is for the manual RNAscope Multiplex Fluorescent Assay. We are very excited to announce that our webinar on the RNAscope LS Multiplex Fluorescent Assay on the LICA-Bond RX system is coming soon. So this is scheduled to be presented on the 13th of September, so please stay tuned for that. Now, this is a very high-level overview of the RNAscope workflow. RNAscope is based on proprietary in situ hybridization technology based on unique probe design and signal amplification system. Now, just as in any in situ assay, RNAscope assay involves permeabilizing properly prepared samples to allow for complementary oligoprobes to bind to the RNA target of interest. Now, as you can see here from the schematic, specific signal is picked up by the double Z probe system and amplified by a series of amplification cascade events. The signal, which is subsequently detected, is visualized as punctate dots and can be quantified. Now, for the multiplex fluorescent assay, the signal can be visualized using a fluorescent microscope. Now, I would like to mention here that we do offer the chromogenic assay as well, and for that, all you need is a simple light microscope to visualize signal. We also have a semi-quantitative scoring system for the quantification of RNAscope signal, and I'll be briefly discussing that in the upcoming slides. Now, thus, through a combination of our unique probe design and proprietary signal amplification system, we are able to present you with an assay that is extremely universal as well as rapid and scalable with the capability of single molecule detection in single cells for virtually any genome, any gene, and any tissue. So how does the technology really work? Let me dig deeper into the probe design. Now, as I mentioned before, RNAscope probe design is the key. As you can see here from the schematic here, we use a double Z probe design. So there are basically three components to this Z. The lower end of region of the Z is complementary to your mRNA, so it directly binds to the mRNA of interest. Now, this lower region contains about 8 to 25 base sequences that are specific to your target. The upper region is basically a 14 base paired tail sequence, and that is a region where the preamplifier binds and the cascade of amplification starts, or it occurs in the top portion of the Z. As you can see, there is a spacer as well in between the base and the upper region, and that allows for flexibility of the probe. Now, why is this so critical? Why are the Zs, as I mentioned before, it's a double Z probe design, paired design. So why are Zs used in pairs? Now, in Zs, in pairs, the upper region of the Zs will provide about 28 bases for the preamplifier to bind right here. Now, both the Zs have to sit or have to bind adjacent to each other on your target region, right here on the lower region. Only when this happens will the top part provide a stable binding region for the preamplifier to bind. So if, for example, there is nonspecific binding and there is just one Z that sits randomly somewhere, it binds nonspecifically. Then what happens is that the pair, the other Z, will not bind next to it. When that event does not happen, or when the two Zs do not come together, there is no stable binding region for the preamplifier. Thus, any nonspecific signal will not get picked up and will not get amplified. So this is how we can come up with an assay. We can offer an assay that is highly specific and sensitive at the same time. Now further, this double Z system offers simplicity of design. Why is that helpful, you ask? Now, let's say, for example, this yellow line is your mRNA of interest. Now, if your RNA is intact, the sample is prepared per our guidelines, the assay will hybridize up to 20 Z probe pairs to your RNA. So essentially, when 20 of these double Zs come together, you can pick up one mRNA copy, which is essentially one dot of our RNAscope signal. On the other hand, let's say you're working on suboptimal samples. It could be clinical, it could be archival samples. There are many reasons why the mRNA in the sample could be degraded. Now, if that happens, it's really, even then, RNAscope can work for you. That's because even when it's chopped up, even when the mRNA is chopped up, the double Z pairs are going to bind. And if the assay is optimized in such a way such that only relatively short probe binding to the target is required to pick up or to allow for signal. Now let me switch gears a little bit and discuss the RNAscope workflow. Now, before I do that, let me introduce the multiplex fluorescent product line. So at this point, we have the capability of detecting up to 3 targets at the same time with our multiplex fluorescent assay. The fluorophores come in wavelengths that are similar to FITC, Psi3, and Psi5. The probes for this assay come in channels 1, 2, and 3, annotated as C1, C2, and C3. So if you would recall the double Z probe design, the top part where the preamplifier binds, that signifies the highly specific tail sequence, which is a C1, C2, or C3. Depending on the tail sequence of the probe, the plexing of the target is decided. Now, in the next few slides, I'll go over in detail on how to combine these probes for your assay. Now, as you can see here from the schematic, our assay, the workflow for the RNAscope Multiplex Fluorescent Assay is represented here. So as you can see, it has 4 amplification steps, and the signal is detected using a labeled probe, which is tethered to a fluorophore. So essentially, we have the double Z probes that bind to the mRNA of interest. To the top part of the double Z, the preamplifier is going to come and bind, and that's where the series of amplification or detection, the cascade starts. And then at the very end, we come in with a labeled probe that is tethered to a fluorophore, and you can then detect signal. So as I mentioned before, the signal can be detected in up to 3 channels, so they can be optimized for Alexa 488, Auto 550, and Auto 647. Just some probe-related pointers here. By default, the C1 probes would be are provided as 1X. They are ready to use, and they are provided in eyedropper bottles. On the other hand, the C2 and the C3 probes are provided in tubes, and they are provided as 50X. Thus, in order to plex, say for example, if you want to detect 3 targets at the same time, you would mix the C2 and the C3 probe in the C1 probe in the ratio of 1:1:50. If, for example, you have targets that are not in the C1 probe, they are in the C2 and or the C3 probe, that's no problem at all because we do provide a probe diluent which you can then use to make your probe mix and use accordingly. This, as you can see here, is represented the workflow of the fluorescent assay. As I mentioned before, it has 4 amplification steps, and the labeled probe, which is tethered to the fluorophore, comes at the very end. So coming back here, as you can basically see here, this represents the workflow for fresh frozen samples. Now, why is that? That's because that fresh frozen is the recommended sample type for the multiplex fluorescent assay, and that is why we have represented that here. Having said that, if you're working on fixed frozen samples or FFPE or cultured cells, please do check our website. We have user manuals to perform the multiplex fluorescent assay on those samples as well. So coming back to the fresh frozen sample type, the pretreatment essentially is very brief. So essentially the tissue undergoes a very, very brief post-fixation in 4% PFA, cold PFA for about 15 minutes, followed by dehydration and protease digestion step. Then we come in with our target probe, which you hybridize for about 2 hours at 40 degrees, followed by washes and the amplification cascade. At the end, you do DAPI counterstaining and image detection under a fluorescent microscope. I would like to point out that pretreatment conditions may vary based on sample type. So if you have any questions at all, please do feel free to reach out to technical support or check our support website. We have a wealth of information there as well. Now, we do provide flexibility in terms of planning your experiment. So our assay is developed such that you can either do the entire assay in one whole day, right from pretreatment all the way to detection and visualization. It can certainly be done in one day. Or if required, you can stagger it and divide it among 2 days as well. So properly prepared and stored samples are pre-treated and put through the entire assay in one day. The other option being stop after pre-treatment and continue with the amplification the next day. Our user manual is actually peppered here and there with optional assay stopping points. So if you have any questions regarding that, please do refer to the user manual. So let's kind of dig deeper and get into the assay. So here are basically some tips for performing the manual multiplex fluorescent assay. And at a very, very high level, there are 4 pointers or 4 critical points that we would request our customers to pay attention to. So those would be the protocols. So as I mentioned before, online we have very detailed sample type specific protocols available for the assay. So please do follow protocols exactly as described in the user manual. Then comes sample pretreatment. Now this is a critical feature, this is a critical aspect that contributes to the success of the assay. So please review sample pretreatment recommendations for your particular sample type. The third aspect is controls. So in controls, we recommend the use of positive and negative control probe, as well as cell pellets, which can be used as a positive control sample. I will be discussing the use of control probes and control sample in greater detail in the upcoming slides. And finally, the checklist. So online, we have a checklist that's available. If you go to our support page and click on Getting Started, you'll be directed to a web page saying checklist. So please do, it's got an interactive drop-down menu. Choose your specific sample type and the assay that you're performing, and you will be provided with a checklist. So I would recommend that our customers go through all that and make sure that you have all the critical must-haves for the assay. Here's what the checklist would look like very briefly. We certainly recommend the use of the image hydrophobic barrier pen. Now, this is the pen that we recommend, as it's been validated time and again by our R&D scientists, to be the pen that holds up a very strong barrier throughout the assay. Use of positive and negative control probes. So for the multiplex fluorescent assay, as I mentioned before, you have the capability of detecting signal in up to 3 channels. Thus, to facilitate that, we offer 3-plex positive and 3-plex negative control probes. In the 3-plex positive control probe, we have 3 different housekeeping targets in the 3 channels, C1, C2, and C3. So, and in the 3-plex negative, we have DAP-B, which is a non-specific bacterial target in all 3 channels. I highly recommend the use of these 3-plex positive control probes, not only as a workflow check, a technique check, but also to ensure quality control of your sample, to ensure that RNA quality is intact, allowing for easy interpretation of results. Now, for our customers that use fixed samples, it could be fixed frozen or FFPE samples, there is a pretreatment step which requires you to do a heat-mediated target retrieval. Please note that this is not part of the pretreatment protocol for fresh frozen samples. For this step, we recommend the use of either a hot plate or a steamer. I will go through the use of the hot plate in greater detail in the upcoming slides. We also recommend the use of superfrost plus slides. Now, our assay has got many different amplification, incubation, washing steps. Thus, we don't want our customers to lose their samples halfway through the assay. Thus, based on extensive R&D testing, we determined that the superfrost plus slides promote optimal sample attachment. Thus, we highly recommend the use of these slides. For hybridization purposes, as you would note, there are a lot of steps that need to be done at 40 degrees Celsius. We recommend the use of the HYBEZ hybridization system, and I'll be covering this in the upcoming slides as well. Run RNAscope control slides. Now, for our customers that work with human samples, we offer HeLa cells. And for our customers that work on mouse samples, we offer 3T3 cell pellets. These are FFPE cells. We recommend the use of these cell pellets along with the positive and negative control probe as a technique check. For mounting, we understand that our customers may have used a lot of different mounting media for other applications, but for the fluorescent assay, for the multiplex fluorescent assay in the RNAscope context, we certainly recommend the use of prolonged gold anti-fade mounting media. And then please do use fresh reagents as applicable. So as I mentioned before, this is a typical hot plate setup, which is required for target retrieval for our fixed sample, the customers that use fixed samples. Now, the most critical points to watch out for here are the temperature of the buffer and duration of the boiling. If either of these parameters are overextended, the tissue may be overdigested. Now, this may also lead to sample detachment. So if these parameters are extended. Now, one suggestion we have here is to make sure that the buffer is at a gentle boil without jets of bubbles shooting up from the bottom of the beaker. If you do see those bubbles, please try to position the slide rack away from the bubbles. Another recommendation would be to cover the beaker with a foil to prevent boiling over. And we also have a steamer protocol in place for this particular step. So if you have any questions, do let us know, and we can send you the protocol for performing target retrieval in a steamer. It's also available in our user manual. So essentially, when you order the RNAscope Multiplex Fluorescent Kit, this is what you're going to be getting. You'll get pre-treatment reagents. Again, please note that these pre-treatment reagents are designed or geared towards fresh frozen samples. You will be getting the RNAscope detection kit with the amplification reagents and the wash buffer. Now, should you be working on fixed samples, please do let your area accounts executive know, and they will certainly help in providing the correct pre-treatment reagents for your particular assay. Some pointers here. Please do note that the probes are warmed at 40 degrees for 10 minutes before use. As for the wash buffer, it's provided at a 50x, so please do warm it for at least 20 minutes at 40 degrees before reconstituting to 1x. Now shown here is our HYBEZ hybridization oven. Now customers who have been in touch with me have heard me talk about this oven very often. Now that's because it has been consistently validated by our scientists to be the very best for our assay. Why is that? That's because this oven has been specifically engineered with our assay in mind and shown to maintain temperature and humidity at its most optimal. As you can see here, the oven comes with a humidity tray, a slide holder, and humidifying sheets. It is critical to moisten those sheets before you run the assay. So do moisten them and slide the humidity tray in the oven even before you run the assay. Now, your success with RNAscope is our top priority. So thus, I can assure you that if you use this oven on properly prepared samples, your assay will certainly be a success. If you have any questions on how to acquire this oven for your needs, please contact your area accounts executive. Now shown here is just some accessories for the washing steps. So many of our customers certainly use the TissueTech wash tray for that. However, in order to streamline the entire wash process and to make it extremely convenient for our customers who run multiple number of slides, we have designed the Easy Batch slide processing system. So as you can see here, there is that white slide rack. These slides, they fit or they kind of slide into these grooves right here. And using these systems at the very end, you pinch them in place and the slides get locked in. Now, the very, very useful aspect about this is that this slide rack goes seamlessly between the wash tray, as shown here, and the humidity tray that I showed before in the previous slide to be used with the HYBEZ oven. So thus this makes the washes and the repeated amplification steps a breeze, because all you need to do is kind of hold it vertically and give it a couple of flicks or smart shakes, and then the excess reagent will get removed. Then it can go into the wash tray, and after the wash is done, you can add the amplification components, and it goes right back to the hybridization oven. So there is no taking one slide out at a time, blotting it out. No, it certainly eliminates the extra steps and a requirement of time. Thus, here, just to summarize, here's what we recommend that our customers follow. Certainly apply all amplification steps in the correct order. The reason why I bring this up is that there are many customers who may be working with only 1 or 2 targets in the multiplex fluorescent assay. Even if that is the case, we certainly recommend the use of use of AMPS 1 through 4, because that's the one that builds the detection tree coming in with the labeled probe at the very end. And as I mentioned before, use the flicking or tapping technique to remove residual reagent. Do not let the slides dry out. Now, you may notice in the user manual that there are very specific parts of the assay where it does call for drying out or dehydration. For example, when you have to draw the barrier using the image pen. So unless and otherwise stated in the user manual, please do not let the slides dry out. Make sure that the hydrophobic barrier remains intact throughout the assay, and please do not alter the protocol in any way. Please do reach out to technical support if you have any questions regarding the suitability of any step for your sample type. Warm the probes and wash buffer at 40 degrees in order to prevent precipitation. Make sure it's entirely homogeneous. Please maintain adequate humidity in the humidity control chamber, and again, please use fresh reagents wherever applicable. So let's shift gears and get into some specifics, and let me show you some examples. Here is what you can do in order to customize the color output of your target. So we would like to give our customers the option of flexibility in terms of channel choice. So keeping that in mind, our fluorescent assay comes with 3 AMP4 alternatives. Now these AMP4FL alternative reagents are allowed for flexibility of switching channels for a target using the same probe without the need to reorder the probes. So let me very briefly walk you through how you can do that, but if you have any questions, please do refer to pages 7 and 8 of the detection user manual. So hypothetically, let's say you have 3 targets. For simplicity's sake, I'm calling them targets 1, 2, and 3, and they are in channels C1, C2, and C3. Let's say you use Alt-A, AMP4 Alt-A. If you do that, target 1 will be visualized in the green channel, target 2 in the orange channel, and target 3 in the far red. If you use Alt-B, 1 and 2 get switched, and if you use Alt-C, then then what happens is target 1 will still be visualized in the orange channel, but 2 and 3 get switched. I would certainly recommend our customers to visit our RNAscope Multiplex Fluorescent Assay webpage for a detailed description of how the technology works. So essentially, the multiplex fluorescent assay provides the same exceptional sensitivity as our singleplex assays, allowing for single molecule detection of up to 3 RNA targets simultaneously. And as I mentioned before, the reagent kit is equipped with reagents to multiplex 3 probes using the FITC, Cy3, and the Cy5 dyes. At this point, if I may, I would like to bring up some terminology which may not be very clear in terms of the channels. So you may have heard me talking about probe channels. So probe channel is nothing but the tail sequence, which is the C1, C2, and the C3, which essentially goes back to the top part of the double V system, and that guides your targets in the plexing pathway. However, there is also the microscope channel or the filter settings. Now, those would be the filter settings that are probably optimized for the detection of Alexa 488, Auto 550, and Auto 647 dyes. So this is what you would optimize, optimize your microscope filter settings for detection of the RNAscope signal. As promised before, let me talk about qualification of samples using our controls. So essentially, shown right here is a schematic which shows in a step-by-step fashion on how to qualify your sample for the sample for RNAscope assay. Now, we recommend that our users perform this whenever they are starting the assay or using a new sample. The first step, as you can see here, is a workflow or a technique check. In order to do so, please run positive and negative control probes on ACD control cell pellets. So depending on the species, we offer HeLa and 3T3 cell pellets. Once this is done, once this, your workflow you ensure that the workflow is fine, you may then move on to your sample. Here, we require we request our customers to do a QC or a quality check of your sample by running the positive and negative control probe. You will be looking for 3 parameters. You'll be looking for the signal intensity based on positive control staining. You'll be looking for background in the negative control slide, and you'll be assessing the efficiency of the pretreatment. The pretreatment protocol that we have put in place is very universal, but there may be certain requirements depending on unique tissue type or sample preparation that the pretreatment that we specify in our user manual may not be most optimal for your tissue. So thus, we recommend that you first run the standard conditions using just the positive and negative control probe, and we can then work with you in assessing if this was optimal for your tissue type, for your sample. Once you're done with this step, you can then move on to running your target probe on the sample. This workflow right here, this flow chart basically just reiterates what we just discussed in terms of sample qualification. So do a technique check. Once that's passed, move on to sample check, an RNA quality check, and once that's passed, move on to your target markers. Now, this then brings me to optimization of the assay. So as I mentioned before, our pre-treatment conditions are universal. They should be applicable to a majority of tissue types and sample preps. However, there may be conditions where optimization may be required. So this slide right here is basically addressing all three sample preps. So essentially, as I mentioned before, fresh frozen is a recommended sample type for the multiplex fluorescent assay. However, let's say you're working on fixed frozen samples, let's say you're working on FFPE samples. As I've laid out here, these are some cases where the fixation may not be optimal. Say, for example, for the fixed frozen, what we recommend is 24 hours, 4 degrees and 4% PSA. If it's less than that, then it's considered underfixed. For FFPE, what we recommend is 10% NBF at room temperature for 16 to 32 hours. Now, if it's more than that, let's say it goes on for 48 hours or more, it's certainly considered overfixed for our assay. In addition to that, there are special sample types, like you have your xenografts, the cultured cells, the cell pellets. So they have specific pre-treatment requirements. We have special tissues. So on the one hand of the spectrum, you have samples like liver and muscle, which may need some extra penetration. And on the other hand, end of the spectrum, we may have sensitive samples like tissues like lymphoid tissues, which may need very, very mild pretreatment. So thus we take all that into consideration, and we've worked with you in determining the most optimal pretreatment for your sample. So these are just some of the factors that affect RNAscope assay performance and some ways to fix them. So if the fixation conditions are not optimal based on the first QC run, you may certainly gain by fixing the samples as recommended. And I do want to mention that in the fresh frozen scenario, there's a very, very brief post-fixation using either 10% NBF or 4% PSA for 15 minutes. In case the RNA is degraded in your sample, we would recommend that you acquire new samples and assess RNA quality. In case hybridization conditions are not optimal, we certainly would recommend the use of the hybridization oven to ensure optimal hybridization conditions. If you find that sample drying out is an issue during the assay, the root cause may be the use of a pen that's not the image hydrophobic pen. So in that case, we would certainly recommend the use of that pen. And as I mentioned, special tissues that are sensitive to pretreatment, the rule of thumb is to certainly start with standard pretreatment conditions. We will review the images with you and then optimize accordingly. So this is how you would optimize, as I mentioned before. Start with standard conditions and observe the staining pattern. Now, perhaps there's high background in your samples. Now, high background may stem from the sample being over-digested, and that may directly be related to the samples being underfixed. Perhaps you are facing issues of very weak signal or no signal at all. Now, that may stem from the fact that the sample may be if there's no signal at all, then the sample may be underdigested, and this may be related to the samples being overfixed to begin with. Now, in order to address these issues, you would either adjust the boiling time. This would certainly be applicable to fixed samples, the fixed frozen and the FFPE samples. For fixed frozen samples, the standard is 5 minutes. For FFPE, the standard is 15 minutes, so you can certainly adjust that. As far as fresh frozen samples go, you can adjust the protease time. The standard is 30 minutes at room temperature. Now, I'd like to mention here that we certainly support the use of the RNAscope Multiplex Fluorescent Assay on cultured cells. The requirement there is the dilution of the protease, and a very good starting point is 1 in 15 in 1x PBS. Now, here's what a successful assay would look like. Now, as I mentioned before, we offer the 3-plex positive and negative control probe, what we look for is the pattern. So essentially, in the 3-plex positive control probe, we have put a relatively low expressing housekeeping target called polar 2A in channel 1, PPIB, which is a medium expressor in channel 2, and UBC, which is a high expressor in channel 3. And as you can see here, this assay was done on the HeLa cells and developed using Alt-A. Thus, you can see a low expressor, polar 2A in the green, medium PPIB in the out of 550, and high expressor in the far red channels. Now, let's discuss some scenarios for troubleshooting. Now, as you can see here, in the top, in the left, top left, we have a fresh frozen mouse kidney that looks highly overdigested. Now, the possibility there is probably that the protease step was done at 40 degrees. For fresh frozen, we recommend room temp, and when it was done at room temperature, as you can see, the signal is much improved. Similarly, this is a case of fresh frozen mouse brain. Same thing, the incubation was done at 40 degrees, and it was very improved when it was done at room temperature. Now, sample detachment is a very commonly observed issue, which is very, very easily addressed. So what I have here is a schematic that touches upon all three sample types. So let me very briefly walk you through that, and if you have any specific questions, please feel free to reach out to us. Now, if you're working on fresh frozen samples, then you would certainly benefit by the use of SuperFrost Plus slides, by sticking to the 10 to 20 micron range of thickness, and including this very, very critical air drying step at -20 degrees. So right after cryosectioning, we recommend that our customers dry their slides at -20 before transitioning to -80. Now this -20 step applies to, now all these three apply for fresh fixed frozen samples as well, but in addition to that, we certainly can include additional post-fixation steps, additional baking steps to address sample detachment. So this would then depend on where, when, and how you observe the detachment, and we'll work with you on that. As you can see here, for FFP and fixed frozen, you can certainly see detachment at various stages, and it could be before the target retrieval, the protease. We would certainly work with you to address those issues. Now here, as I mentioned before, the recommended thickness is about 10 to 20 microns. So sample thickness can certainly affect the signal in your sample, because that has got a direct bearing on how efficient the pretreatment was. So as you can see here, 10 to 20 micron range gives very pretty robust RNAscope signal. Now, tissue autofluorescence is a potential issue with this particular assay when fixed samples are used. Now, as I mentioned, fresh frozen is certainly the recommended sample type, and the multiplex fluorescent assay may not pose the autofluorescence issue if you're working on the fresh frozen samples. So here are some examples of FFPE samples that have the problem of tissue autofluorescence. And again, we have ways to address that. In-house, we use background correction software to distinguish the autofluorescence from the real signal. We can provide those recommendations if you have any questions about that. And we also recommend the use of the AMP4 alternatives that we have provided. So you can, if you see, let's say for example, you see autofluorescence in the Alexa 488 channel, then you can certainly use those AMP4s to switch the color module, and that we have seen can certainly address the autofluorescence issue. Now, very, very briefly, the troubleshooting issues, or the issues that our customers face with the multiplex fluorescent assay, come from two different angles. There's microscopy related and sample prep related. Now, if it's microscopy related, let's say there's weak signal, non-specific signal, it could perhaps be stemming from the use of wrong filter settings, use of longer emission cutoff, using the wrong exposure, or inappropriate imaging. Now, all these can very easily be fixed by the use of correct filter settings. So as I mentioned before, optimized for Alexa 488, Auto 550, and Auto 647. And we also recommend not using the auto exposure at first. Please do verify the signal with naked eye before moving on to auto exposure. And again, use known image enhancing software. So as I mentioned before, in-house we use a software called Nuance from Perkin Elmer. Now, sample prep, again, that's one of the areas that may contribute to suboptimal signal, but very, very easily addressed by preparing samples according to our guidelines. So if you see no signal, weak signal, perhaps that could be because of compromised RNA quality, as well as sample preparation. So let's say there's high autofluorescence in your fixed samples. This is very easily addressed by switching to the fresh frozen sample type. So thus use new sample with good RNA quality. Follow the pretreatment guidelines that we recommend. Always to perform the 3-plex positive and negative control probe, and please check signal with naked eye under the objective lens first. Now, I'll be very happy to take any questions that you have, but let me just address these very, very commonly or frequently asked questions. Now, assay compatibility, as I mentioned before, fresh frozen is the recommended sample type, but we certainly support the performance of RNAscope Multiplex Fluorescent Assay on FFPE samples, on fresh frozen samples, on fixed frozen samples, tissue, as well as cell pellets, cultured cells, primary cells, and so on. One of the key differences between the RNAscope ISH and IHC in case of FFPE samples would certainly be in the target retrieval step. There is no cooling required during epitope retrieval in RNAscope. The user should directly put the slides in water at room temperature, dehydrate, and proceed to pretreatment. At this point, if you have any questions, I'll be happy to address them. Now that we have a couple of minutes, I'd like to take some time to walk you through our website. So as you can see here, our support page has a wealth of information. So I would certainly recommend taking a test drive to our website. So as you can see, if you place the cursor over support, you get a drop-down menu. So let's go to Get Started first. And as I mentioned, the pre-run checklist. So there's a pre-run checklist that goes that takes into consideration your sample type, your assay, and then it kicks out a checklist that is required for you to successfully perform the assay. So let's just quickly do fresh frozen for the multiplex fluorescent. So in the first drop-down, you would choose manual 2.5 HD because that's our current newest version. For sample type, you'd use fresh frozen, and for detection, you'd use the multiplex fluorescent reagent kit. When you hit search, you are then presented with a personalized, customized checklist very specific for your needs. So I'd certainly recommend that you do take a look at that. And this Getting Started page has information on tissue and sample prep, a very, very high-level overview, as well as other details to consider for the assay. Now, as far as user manuals go, again, because we offer two different types of assays, we offer the fluorescent, the chromogenic. In the chromogenic, we offer different flavors. And on top of that, we support so many different sample types. We have a lot of different user manuals available online. So again, you would choose the user manual that works the best for you or that's best applicable for you. So use manual 2.5 HD. For sample type, again, depending on what it is, so let's do fixed frozen now, and detection multiplex fluorescent. And when you hit search, you are presented with 4 results. So the first one talks about sample preparation. So for example, in fixed frozen, we recommend that the sample be put through the sucrose gradient, fixed for 24 hours with 4% PFA, all those details. In addition, it talks about the specific pretreatment protocol. And then the part 2 right here picks up from probe hybridization and takes you all the way to amplification and detection. I would like to thank you all for attending today's webinar. If you have any questions, please feel free to reach out to us at technical support. You may either email us or call us anytime. Again, thank you for attending the webinar. Have a nice day.